Cambridge, after the war, was a city of battle-weary academics and students who'd already seen the harsh side of life. This small university city was about to be the centre for not one, but two of the most significant discoveries ever made. On February the 28th, 1953, two rather wild young men burst into this pub in Cambridge, the Eagle, and announced to anyone who would listen that they had discovered the secret of life. What they discovered was the structure of DNA, the genetic molecule, the physical trace of the universal database of all life. It was more than just a structure. Written all over it was what it does. It has the extraordinary capacity to make almost exact copies of itself. Like every biologist of my generation, I'm hugely indebted to those wild young men and their truly inspirational research. They were an unlikely pair, not what Cambridge was used to. The brilliant, drivingly ambitious young American, Jim Watson, and his clever, larger-than-life English colleague, the garrulous Francis Crick. They were irreverent, arrogant even, searching for nothing less than the secret of life, what Watson himself referred to as the most important event in biology since Darwin. Nearly 100 years earlier, Charles Darwin had published his great book, The Origin of Species, and the theory of evolution broke upon the world. But while Darwin's theory explained how life had evolved, one key piece of explanation was missing. How and why was this information passed down from parent to child? That is the secret of life, and for a generation that had just gone through a devastating world war, solving this mystery was one of the great challenges facing science. In this quest, Crick and Watson had one great advantage: each other.
If, for example, I had some idea, which, as it turned out, would say be quite wrong, was going off at a tangent, Watson would tell me, in no uncertain terms, this was nonsense.
Well, Francis likes to talk. It's his dominant quality, I think. He doesn't stop unless he gets tired or he thinks the idea is no good.
It's useless working with somebody who's either much too junior than yourself or much too senior, because then politeness creeps in. And this is the end of all real collaboration in science.
Crick and Watson understood each other. That was essential. But the problem before them was immense. For some decades, scientists had thought that a substance called DNA, found in all living cells, might play a key role in transferring information from one generation to another. But no one knew how it could do this. Perhaps if they could see into its structure, they might be able to discover how it worked. But looking closely at DNA was very difficult. Direct visualization, as with a microscope, couldn't work. The scale was too small. But there was an indirect method involving X-rays, and X-rays were already being beamed at crystals of DNA just 50 miles away in London.
She is one of the heroines of British science, sometimes called the Dark Lady of DNA. Her name was Rosalind Franklin. A young woman in the then male-dominated world of King's College London, she, like Crick and Watson, was trying to visualize the structure of DNA. And Rosalind had skill on her side. She was an expert in X-ray diffraction, a way of capturing images of atomic structure. But the kit she had to work with was incredibly basic.
The new biophysics department at King's was a pretty ramshackle place. A lot of the equipment was borrowed from colleagues at other London colleges, and this camera was army surplus. The DNA came from the thymus gland of a calf in Switzerland, And in order to take pictures of it, it had to be suspended on a paperclip. You put it in here, you look through the eyepiece and line it up to make sure it's straight. Then you put a piece of photographic paper here and bombard it with X-rays. The X-rays hit the crystal and scatter the light, giving you a pattern of dots. If the image is sharp enough and the sample was lined up properly, you might be able to work out what the structure of the crystal is.
Rosalind was extremely determined. Little would stop her. The X-rays were done at night, when there weren't many people around, because King's College was afraid there might be leaks from the lab. But it wasn't Rosalind who made the first breakthrough. It was her colleague, Maurice Wilkins.
Early in 1951, Maurice Wilkins took a photograph of DNA that suggested it had a regular structure. The story goes that one night he came outside for a breath of fresh air and looked across the river at the Oxo Tower.
The famous logo gave him an idea. Perhaps the structure in the picture was shaped like an X, which might mean DNA was a helix or spiral shape. This was a vital insight, and perhaps, if they had been able to work together, Maurice and Rosalind could between them have begun to crack the mystery. But that was never going to happen.
Rosalind comes across as a very private person, a loyal and thoughtful friend, very honest, a brilliant scientist and a dedicated one. But she also had a fierce temper and contempt for those she didn't respect. She refused to collaborate with Wilkins and didn't show him her data.
So when she took this photo, she didn't show it to Wilkins, or indeed anyone else. This is the famous photo 51, a blurry image, but one which holds the key to the secret of life. In this picture are vital clues needed to discover the shape of DNA. But Rosalind Franklin tucked the picture away in a drawer. And there it might have stayed, if it wasn't for Wilkins. Over 8 months later, in January 1953, James Watson made one of his occasional visits to King's. Wilkins, who worked in the same lab as Rosalind, said he had something to show him.
For Watson and Crick, this was the crucial piece of evidence, the clue that would allow them to deduce the structure of DNA. The pattern of spots on photo 51 showed clearly that DNA was a helix.
Watson saw immediately the value of this information. He hot-footed it back to Cambridge with this vital clue. But it's what he and Crick did next that made all the difference. They knew it was a helix, but now the question was, what sort of helix? Their approach was very hands-on, making giant 3D models of all the different shapes they could think of that might fit the picture. They tried model after model, each one a hypothesis for a three-dimensional structure that might explain Franklin's X-ray results. Watson and Crick worked at a furious rate. The machine shop, which supplied them with the tin bases for their models, struggled to keep up. But the pieces of the puzzle all fell into place in February 1953. Rosalind Franklin's X-ray pictures and her measurements on them enabled Watson and Crick to work out that DNA had to be a double helix. Their original model has unfortunately been destroyed. This is a half-scale replica. You can see the two spirals going around like that, forming a kind of spiral staircase. And the steps of the spiral staircase are the so-called bases. There are only 4 kinds of them, and they are in pairs. Each step is one pair. The shape plays a vital role in the way DNA works, because the double helix can split perfectly and then reform. The reason we're all different is that we're born with our own particular sequence of these base pairs, with half of each pair inherited from each of our parents. Crick and Watson had indeed made the first step to uncovering the secret of life.
They'd done it. Surely they'd had a lot of luck, but let's not take away from their achievement. As Crick himself said, it's true that by blundering about we stumbled on gold, but we were looking for gold. And the biggest nugget of all? Forget all mystical ideas of an essence of life or a life force. From now on, the essence of life was just bytes and bytes and bytes of digital information.
But the story didn't end there. What's perhaps even more exciting than what Watson and Crick did in the 1950s was what Crick went on to do in the 1960s, cracking the genetic code itself. Crick knew that DNA was information, a code for life. A message lurked in the sequence of DNA bases, which was translated into a corresponding sequence in a different kind of molecule, protein. There must be a dictionary, a lookup table from DNA sequences to protein sequences. That was the elusive genetic code, and that was what Crick set out to break. And what he came up with was an ingenious experiment. He began with a virus and then mutated its DNA. He found that if he removed a single base, the virus would no longer work. This meant the original message the virus DNA was carrying was disrupted. The same was true when he removed 2 bases. If he removed 3 bases, however, the original message was restored. The virus was back to normal. It now remained for others to fill in exactly what each triplet means. But perhaps the vital step in the whole molecular genetic revolution was Crick's discovery that it was a triplet code. It opened up a whole new world of understanding that couldn't have been dreamed of before Crick and his colleagues came on the scene. Since this great discovery, the advances have come thick and fast. We now have a biotech industry worth billions. We can identify medical problems in the womb, identify crime suspects using unique DNA fingerprints, sequence the entire human genome, and even clone new life.